everyone. We are doing a biotechnology series in which we have been discussing various technologies related to the biological systems that we can harness for our own benefit. In the biotechnology, we have discussed recombinant DNA technologies and other advanced genomic and other technologies. Today in this session, I, Dr. Charu Dogra Rawat from Ramjas College, University of Delhi, would be discussing about the next generation CRISPR technologies and, if the time permits, the applications in agriculture. We had discussed, so the important learning objectives for today's session would be to understand next generation or alternative gene editing technologies and to understand various applications of CRISPR-Cas9 gene editing tool. We start with in the agriculture, where we talk about the crop improvement, agricultural breeding technologies and domestication of wild species. Now, before I go further, let's talk about, let's review what these CRISPR-Cas9 system was. So, CRISPR is clustered regularly interspaced short palindromic repeat sequences, and Cas9 is the protein that associates with this CRISPR element and then conducts various kind of editing in the genomes. This is the ideal CRISPR locus we have already talked about, and you can refer to our previous sessions. But just to briefly quickly review, this is the CRISPR locus, which contains the direct repeats, which is shown as blue blocks here, and then there are protospacer elements, which is basically different sequences. Now, this CRISPR naturally is because of the immunity, it plays a role in the immunity that it gives to the bacteria. For example, the introduction of any exogenous DNA, a small piece of exogenous DNA is kept in between these directed repeats as protospacer elements. So, this remains as the memory. So, when it again does infect the bacterial cell, then because of this sequence, it can combine, it can transcribe into mRNA, and then that RNA combines with the Cas9 proteins, where we have this kind of a cluster of Cas9 proteins, and it can basically combine with that and then remove or degrade these exogenous DNA. So, this is the mechanism of action. So, we have got these interspersed repeats in between, and there are direct repeats, and then there are different protospacer sequences. These protospacer sequences are a memory of any kind of exogenous DNA, for example, shown here as a bacteriophage infection and a bacteriophage DNA, and then there are Cas proteins. So, they are translated in the sequences are translated into an RNA, and then they are basically broken down into different elements, these kind of 20 base pairs, around 20 to 50 base pair, these kind of structures by tracer RNA. It combines with the Cas9, and because it shows some homology with the incoming DNA, it binds with that and create a double stranded break. So, this is again showing the same thing, but in a detailed manner. Then when the target DNA, you have a homologous sequence in these spacer sequences, then this is transcribed into an RNA, which is then tagged by these tracer RNAs, where they can be cut into these kind of CRISPR elements, and these elements then combine with the Cas9 protein, and because of the homology that it is carrying, because of this spacer sequence, it creates it combines, it is directed, it is guided towards this target, and then this Cas9 protein, which is a nuclease, creates a double stranded break. Now, the requirement is this PAM motif, which is generally NGG, for example, used with the Cas9 protein. So, this PAM is a requirement. So, any sequence adjacent, any target sequence adjacent to this PAM motif, if it is contained in that kind of a protospacer element, then it can be basically cleaved to create a double stranded break. Now, this double stranded break can be repaired within the cell, and therefore, if we design these guide RNAs, that is guiding this element onto the target RNA, then practically, theoretically and practically, we can target any target, and therefore, it can create a double stranded break. So, this is the mechanism of action of a Cas9, CRISPR-Cas9 system that we talked in very much detail in our previous sessions. However, the reliance on the DSBs, which is the double-stranded breaks, to stimulate the gene editing process may undermine its safety. Now, double-stranded breaks can delete thousands of base pairs and create new genotypes, and they may have potential pathogenic consequences in mitotically active cells. So, this is one of the disadvantage of creating the double-stranded breaks, which is a prerequisite for gene editing in the basic CRISPR-Cas9 technology. As you know, many bases can be deleted, creating new genotypes, and probably it can revert to a lethal arrangement, which can basically be pathogenic for the mitotically active cells. For instance, CRISPR-Cas9 genome engineering in human pluripotent stem cells can generate p53 mutations, limiting the potential application for cell replacement therapy. So, this is one example. Now, p53 is a very important molecule and plays role in various kinds of cell signaling pathways. So, if this p53 mutation is generated, then the cell replacement therapy is has a very limiting potential because of that, because of this kind of a double-stranded break. High frequency of indel mutation limits the efficiency and the incidence of CRISPR mediated HDR as observed in HEK293 T cells, which is only 38%. So, again the indel mutation, as we discussed in pluripotent stem cells, these kind of mutations they can limit the efficiency and the incidence of CRISPR mediated homology direct repeats that we had talked about in the mechanism of action, how the cell repairs. So, this is a limitation of creating a double stranded break. Alternative mechanisms such as deaminase mediated base editing and reverse transcriptase mediated prime editing, alternative CRISPR-Cas based genome editing technologies have been developed. These technologies do not involve DSB formation, they do not require donor DNA, and they induce precise sequence editing and are more efficient than the homology directed repeats. So, these next generation CRISPR technologies are the recent and very advanced technologies that has been developed and utilized for creating genome modifications. As we said that the major thing was the creation of the double stranded break. Now, these technologies, which we will talk in detail, the double stranded breaks are not formed, and also it does not require any kind of a donor DNA. So, let us look at that. See, the idea of double stranded break is, or when we say insertions or deletions, it is generally the modification of the base, because the A, G, C, T in the DNA gets modified or replaced or substituted by another base. So, if we do not create these kind of double stranded breaks and still cause the base editing to be done, then it will counter the limitations that we are facing with the CRISPR-Cas9 technology and the harmful repercussions that the double stranded breaks are creating in the cells. So, one of the technology is the base editing technology. Now, base editors, they comprise nuclease impaired Cas9 fused with the deaminases. So, nuclease impaired, because then the Cas9 was the one which was creating a double stranded break, but if the nuclease activity is impaired, then the Cas9 will not be able to create the double stranded break. And these Cas9 molecules are fused with the deaminases, which will cause the base editing to be done, the substitutions, etc. Currently, there are two major types of DNA base editors, the cytosine base editor CBE and the adenine base editor or ABE. They can mediate all four possible conversion mutations, that is the base C to T, A to G, T to C and G to A. So, they are converting, they are making these base changes caused by these kind of cytosines and adenosines, deaminases, and then the Cas9, the CRISPR thing is also guiding it to the target. So, this become a very precise mechanism of gene editing that has occurred. So, let us look at the mechanism of action of these two types of base editing in a little bit of detail. The first one is the cytosine base editing. Now, CBE is composed of a Cas9 nickase So we call this as nickase when it is cleavage property is removed. So this Cas9 nickase referred and written as N-Cas9 bearing the DNA mutation, so that it deactivates the RUVC, one of the two Cas9 nuclease domains. So this impairment of the Cas9 nuclease domain render its nuclease activity inactive. So because of the DNA mutation in this particular gene, the RUVC, which is the nuclease domain, the Cas9 no longer is able to cleave or create the double-stranded breaks. This is fused with two proteins. One is a cytidine deaminase and an uracil DNA glycosylase inhibitor, which is a UDG or inhibitor, which is called as the UGI. So we will see what their roles are or how they act. So the basic thing is that it is a variant of a Cas9 which has been created by mutating the D10A at the D10A position, inactivating the RUVC. Now what the CBE does is that it introduces the C or G and is to T is to A. So this kind of base pair base transition occurs directly into the sites targeted by the single guide RNA. So you can see this is a similar kind of a thing like CRISPR-Cas9, but the major thing or the addition is that this Cas9 is a variant and it is fused with the deaminase protein molecule, as well as it carries this UGI inhibitor. So this also, because it is carrying this guide RNA, this guide RNA is going to guide it to the target where this transition needs to be facilitated. So it goes and binds there, and because of this activity, this will be directly converted into the, so the deaminase deaminates the cytidines to uridines in the non-target strand, which is the single strand DNA part of the R loop generated by the N-Cas9, which is the N-Cas9, which is mutated at D10A position, sgRNA complex, while the UGI prevents the UDG from deaminating cytidines to apyrimidinic sites. So this is an important part. So what does the role of deaminase? So the CRISPR-Cas9 variant N-Cas9 complex, because it contains an sgRNA, which is a single guide RNA, it guides the complex onto the target DNA. In the non-target strand, the deaminase deaminates the cytidine to uridine. So C is first transitioned into uridine, and this is done because this non-target strand is in the R loop generated by the N-Cas9, while the UGI, which is the the inhibitor for UDG, it forms the deaminating cytidine to apyrimidine. It prevents that, inhibits that. Now when this Cas9 induces a nick on the target strand, the DNA mismatch repair pathway or other DNA repair pathways, they are activated and it preferentially resolves the UG mismatch into the desired UA. Following the DNA replication, a T is to A product is generated. So the first thing is that in the cytosine, where the cytosine is there, a uridine is added, and this uridine, because the nick is created, so when it binds to the other one, then this U is resolved to form the G1, and in the next cycle, because this UG is now a mismatch, because instead of C there is a U, and now UG is a mismatch, so it resolves this mismatch into U and A, and in the next sign of replication, this U will be converted into T. So there is a transition that has occurred from C is to G, CG to TA transition has occurred. So this is how the base editing occurs. So deaminases deaminate the cytidines to uridines, and then the inhibitor basically inhibits the deamination on the other strand, and then when they combine, when they because this is a mismatch, it is recognized by the DNA repair pathways, and then in the next cycle of and it is repaired, and in the next cycle of DNA replication, the base transition occurs. So this was the cytosine base editing. Similarly, there is something which is called as the adenosine base editing, and in adenosine or adenine base editing, this expands the base editing to include the AT transitions to GC transitions. So, GC transitions have been done to AT transitions, or basically C is to G transition is converted into T into A type transitions, and now AT can be converted into GC transition or substitutions using what is called as an adenosine deaminase as an effector and fused with the Cas9. So, instead of the cytidine or cytidine deaminase, the adenosine deaminase is fused with the nicase, the Cas9 nicase. So, please remember these terminologies as well, and the symbols, that this Cas9 is a nicase Cas9, where there is a mutation at 10, where the D is converted into A amino acid, and therefore, it loses its property to create the double stranded breaks in the target DNA. Now, adenosine deaminase deaminates the adenosines to the inosines. So, in the first step, they deaminate the adenosines to inosines, which are then recognized as guanosines by the DNA polymerase during DNA repair and replication. So, when this is recognized, so the A will be converted into I, and then that I will be recognized during the replication for the complementarity base pairing. It will be recognized as G, so a C will be incorporated, and this IC will be considered as an error or a mismatch, and which will be repaired by the repair pathways, and then the I will be converted into a C, and therefore the transition has occurred. So, these CBE and ABE methods, these are the base editing methods employing the CRISPR-Cas9 editing tool, but in a modified manner, so that the double strand breaks are not created. So, that is basically the summary of what we have discussed till now. There is another method, an advanced technology, which is the dual base editing. It is also referred to as the saturated targeted endogenous mutagenesis editor or STEAM, and this STEAM, and basically there is a fusion between the cytosine and adenine dual base editor, which is created to simultaneously perform the CG to TA and AT to GC editing in plants using a single guide RNA. So, a single guide RNA is used, and this single guide RNA is used to create both kind of mutations. Why? Because this nicase Cas9 will now be associated with the both CBE as well as ABE editors. So, this is how basically it looks like. So, this is the Cas9, and now they have a fusion protein, a cytidine deaminase, an adenosine deaminase, the Cas9 and a UGI fusion. So, this kind of a tool is developed. So, the base is the CRISPR-Cas9 because the mechanism of action is basically the CRISPR-Cas9 thing, the targeting is in the same manner. So, this Cas9 is now nicase Cas9 is fused with the cytidine deaminase and an adenosine deaminase. So, causing a dual base editing action, and of course, a UGI platform which is there. This STEAM system deaminates the cytidines to uridine and adenosines to inosines in the editing window of the protospacer, and these are then copied by DNA repair and replication, generating the dual CG to 2A and AT to GC substitutions. So, I hope you understand the individual CBE and ABE mechanisms, and when these two proteins, the deaminases, are fused together with the nicked Cas9 protein, then they generate this kind of a STEAM system, and STEAM refers to the saturated targeted endogenous mutagenesis editor. So, where the alternate transition, the CG to TA and AT to GC transition or substitutions occur. Now, another variant of the Cas9 has been generated, which identify the protospacer motif as NG and not NGG. So, we studied the normal Cas9 recognizes the target sequences only when they are adjacent to a motif which is an NG motif. So, again to increase the precision editing and basically to increase the versatility, as many targets they can they can basically edit, a variant has been created termed as the SP-Cas9 NG-PAM variant 127, which recognizes the NG-PAMs and it expands the editing scope and increases the ability to edit as many targets as possible. So, this is just one of the variant I am talking about, but there are many orthologs and other variants that have been developed for Cas9, which is increasing the repertoire of this Cas9 editing tools, and therefore, widespread as well as precise gene editing can now be achieved. These dual base editors, they facilitate the directed evolution of endogenous plant genes in situ. So, because we will be discussing about the application in plants, these dual base editors have been developed for the plants and they facilitate what is termed as the directed evolution. So, when we want one thing to be converted into another and to another and to another, so this is called as a directional evolution, when one give rise to the other by the replacement of the first one, and this directed evolution can be designed and created in situ. So, it has already been done. It might also be used to change the cis elements in regulatory regions and genome-wide screening in a high throughput manner in plants. So, you know that various genes are regulated by various kind of regulatory elements. We had discussed this in our molecular biology series, where we have talked about all different kind of regulations in prokaryotes as well as in eukaryotes. So, the cis elements are when they are present on the same DNA. So, these regulatory regions can also be changed so as to facilitate the difference in the transcription levels of the genes that they are regulating, and again creating the editing that is basically required. Apart from these CBE, ABE and then the dual editors, there is one which is called as a CBE based precise DNA deletion. So, it uses what is called as an APOBAC, Cas9 fusion induced deletion systems. They are termed as AFIDs. These AFIDs contain a certain protein which is like the APOBAC proteins and the Cas9 fusions, and they induce the deletion systems. They are developed to induce the specific deletions within the protospaces. So, like CBE and ABE were creating the base transitions, this is the one which is causing the deletions to occur. They comprise a cytidine deaminase, a Cas9 protein, a UDG and an AP lyase. So, this is the molecule that it contains. Now, how does it works? Cytidine deaminase, it converts the cytidine to uridine just like it does in the CBE system, and then what happens, a uracil DNA glycosylase fused to the cytidine deaminase Cas9 complex excises the uridine and generates an apyrimidinic site, which is nicked by the separate yet co-expressed AP lyase. So, what is happening here is that a pyrimidinic site is created, and this apyrimidinic site is because there is a fusion of uracil DNA glycosylase. If you remember, then in the CBE, we were using a UDG inhibitor in that system, because that inhibitor was inhibiting the glycosylase activity to create the apyrimidinic site. So, there we were preventing the creation of apyrimidinic site because we want that one base needs to be transitioned to another. But here, because we want to delete the bases, we induce the creation of this API site by fusing a uracil DNA glycosylase with a cytidine deaminase. So, the cytidine deaminase works just like in the CBE system, But this uracil DNA glycosylase then excises the uridine that has been incorporated to generate the AP site, which is then nicked by the separate and co-expressed AP lyase. Following this base accession repair, precise deletion is generated between the site of the Cas9 induced DNA DSB and the deaminated cytidine. Now, please notice that we are using the Cas9 protein in this case and not the nicked Cas9. The nicked Cas9 was not able to create a double-stranded break, but in here we are basically using a same Cas9 protein or a wild type Cas9 protein which can create a double-stranded break as well. So, what happens is that an AP site is created and there is a double-stranded break that is present, and therefore, the typical base accession repair mechanism system will be activated and repair it. So, this is again showing the same thing. Now, we have this kind of a protein molecule, and it is this AFID system is going to create the double-stranded break, as well as it is therefore going to remove this particular fragment. So, between the cytidine, wherever this change from C to U has occurred, and then the AP site has been created. So, there is an AP site and there is a double-stranded break. So, it will remove that fragment and will cause the deletion to occur. So, this is again a CBE based deletion method, which is again a advanced or a next generation sequencing, next generation CRISPR technology. Now, although these CBE and AB methods can induce precise base transition, tools for generating base transversions are limited. Now, gene editing does not just include the transitions to occur, but sometimes it also requires the transversions, as it occurs naturally also when the mutations occur. So, for this in 2019, a major discovery system was developed, which was the development of prime editing CRISPR technology. These prime editor technology, it is produced, this technology produce in human cells all 12 kinds of base substitutions, precise insertions or of up to 44 base pair, deletions of up to 80 base pairs, and a combinations of all these cells. So, these prime editing technology or the prime editor technology, it produces various kind of substitutions, insertions, deletions, and all kind of combinations that has occurred. So, this primer editing technology was an important technology that could help in creating the transversions as well. Now, what does this prime editor use? This prime editor uses a fusion of nicase Cas9. Now, this nicase is developed by a different mutation and a reverse transcriptase enzyme, which is complexed with a prime editing guide RNA. This prime editing guide RNA is composed of a reverse transcriptase template and a prime binding site at the 3' end of the sgRNA. Now, we will talk about this prime editing mechanism in detail in our next session. Thank you.